Okay, so this is chapter 19, which is titled The Evolution of Populations. And so we know that all life on Earth is related, and we know that life started once on our planet, and that life has been evolving ever since then, so for millions of years. And evolution over time has shaped each species that either has existed on our planet in the past and has become extinct, or still exists on the planet today. Okay, so we know that life started once, not multiple times, and that since then life has been evolving into the various species that we have on the planet today. We know that the mechanism behind evolution, what's driving evolution, is called natural selection. And we learned a little bit about this in the last chapter. Remember that Darwin is the father of evolution, and him and Wallace both came to the same hypothesis that's been accepted, which again is called natural selection. And what natural selection states is that traits or behaviors, so phenotypes, which are driven by genotypes, there has to be a genetic component to this, that those traits that are adaptive, that make that organism better suited for a particular environment, those traits will be selected for, and therefore those genotypes are selected for. When we say selected for, what we mean is that the environment is going to select those organisms to live. So it's going to enhance their chances of survival, and more importantly, the latter part of this is reproductive success. Okay, so natural selection, again, is going to be selecting for traits and therefore those organisms in a population. And again, that organism is going to have a higher chance of survival and a higher chance, therefore, of reproductive success. It's all about reproduction. It's all about getting your traits into the next generation. That's going to drive evolution. And again, it's important to know that these adaptations, right, these things that are being selected for, they have to be present in the population, right? They're not going to be gained during one's lifetime. Instead, they're going to be things, behaviors or traits that are present in that population to begin with, and that are genetic in nature. Okay, so that, again, they can be passed from parent to offspring. So we say that natural selection, it doesn't create phenotypes. Instead, it selects for adaptive phenotypes, and it selects against phenotypes that are not adaptive. Okay. Remember, in order for natural selection to work, let me just get my highlighter here. In order for natural selection to work, there has to be variation in a population, right? There has to be variation for natural selection to act upon. And so therefore, there's going to be different traits, and those traits that are most adaptive, those get selected for. And again, those traits that are not adaptive, they get selected against and possibly eliminated over time. Where does this variation come from? Where do these new and different phenotypes come from? They're going to come from mutations. Okay, so remember, mutations are the ultimate source of all alleles, right? All of the variations that we see in genotypes originally come from mutations. And just remember, too, that sexual reproduction adds a layer on top of mutations that gives us new and different phenotypes. So sexual reproduction definitely adds to variation in organisms that are capable of sexual reproduction. Okay, so there's this thing called the modern synthesis. The modern synthesis, it integrates, puts together all the pieces of the puzzle. So everything that we've learned about genetics and everything we've learned about evolution, they come together. And so we say that the work of Gregor Mendel has been integrated with the work of Darwin and Wallace. It was finalized in the 1940s, and it is accepted still to this day. And again, it's going to describe how evolutionary processes, so natural selection, can affect a population's genetic makeup, so a population's gene pool, we say, and how this results in the gradual evolution of populations and therefore species. Okay, so we're putting together Darwin and Wallace's work of evolution with Darwin's work of genetics. And now it all makes sense. And so remember, again, we have evolution really on two scales. So microevolutionary changes, as you can see by the name, micro means small changes. These are small changes that occur just over a few generations, right? That are going to change the population's genetic makeup. And we're going to really focus on that in this chapter. And then macroevolution, these are larger scale changes. We'll talk about this just a little bit at the end of the chapter. This is where you have large changes, large evolutionary events, and those lead to the production of new species, okay? Or maybe even higher taxonomic groups, like a new genus or a new phylum, a new class, so on and so forth. Okay, now microevolutionary changes, as they add up in space and time, they will lead to macroevolution. So you can't really differentiate them completely. Okay, your textbook, I like this example because it's a virus, and right now we're going through a terrible viral pandemic. This time it happens to be coronavirus, but it was actually predicted that it would probably be the flu that created this pandemic. Back in 1918, you probably have read about the Spanish flu. The Spanish flu was a pandemic. It hit the United States. It killed a lot of people. Not too long ago, we had the swine flu, the H1N1 flu. And that again killed a lot of people. The flu itself kills a lot of people every year. Flu is an example of a really quickly evolving virus. So when we talk about evolution, again, sometimes it's hard to put wrap our head around because it's hard to see. And yes, we do see microevolutionary changes, and there are scientists that are working on looking at those. But the flu is a really great example of a virus, so an organism. Whether it's living or not living, that's up for debate, which evolves really, really rapidly. And so it's really quick. Its genetic material just tends to mutate really, really fast. And therefore, it evolves really fast. And so when you go to get your flu vaccine, what you're actually getting is you're getting a scientist's prediction of what flu proteins on the surface of the flu virus will look like that year. And there's multiple strains, and so scientists are working with all of those different strains and making predictions about all of those different strains. So, when you hear that the flu vaccine is only 30% effective, a lot of people think that that's bad, but it's actually not so bad, right? They're predicting evolution, which is not the easiest thing to predict. And so if it's 30% protective, even if you get the flu, if it's not 100% effective, you're still going to probably have a milder case of the flu and probably not die, which is great. The same thing now, scientists are working on a vaccine for coronavirus, and so they're looking at what about this virus will stimulate the immune system enough to make memory. And so if you come in contact with that virus, your immune system's already primed, it's already been engaged, you already have memory, and so therefore your immune cells will respond to that virus, quickly and effectively. And so what they're doing is they're looking at surface proteins to try to figure out how exactly they could build memory in our immune system. And so it's a prediction. That work on the coronavirus vaccine is happening right now, and obviously work with the flu vaccine is done every year. One thing that you guys have all been watching the news probably, hopefully you've read some more scientific articles as well, and you've probably heard scientists talking about this, is that we're really not sure what's going to happen with this virus in the future. So is it going to behave like flu? Is it going to be seasonal? And is it going to mutate? Those are all really serious, really difficult questions at this time that scientists really don't have an answer to. So, okay, enough with coronavirus. Let's talk about population evolution. So we know about genetics, right? We did a couple of chapters in the beginning of the semester. We really talked about genetics. We went over DNA. We understand what it codes for. Let's go back and just remind ourselves what a gene is before we get into population genetics. So a gene is going to be a location on a chromosome where there is a specific sequence of DNA, right? A specific sequence of bases that code for an RNA molecule, which in turn codes for a protein, which in turn gives us a specific phenotype, right? Okay, that's a gene. It's a location on a chromosome. And we know that there are different forms of genes, which are called alleles, right? So if you think about eye color, for example, eye color is the gene, and the alleles might be, let's say, pigmented, brown, and non-pigmented, lighter eyes, right? Not brown eyes. And so sometimes there's just one allele for a particular gene, and that's it. Everybody has the same allele. And sometimes there are two alleles, and sometimes there are 3 or 4, right? There could be multiple alleles for a given gene in a population. Humans, okay, most of the genetics problems that we do are they're going to be human related or animal related. And so humans are animals, and all other animals as well are going to be diploid organisms. So what does that mean again? We are diploid, so we inherit 2 alleles for each gene, right? One from your mom and one from your dad. Right? So you get one set of chromosomes from mom and one set of chromosomes from dad. And so you end up with 2 sets, which means if you look at each gene, you have 2 of each gene, or a set, right, of alleles for each gene. Okay, so 2 alleles for every gene. So population genetics. This is a field in biology, a field of genetics, so a subset of genetics, that started in the early 20th century. And geneticists here are going to study how natural selective forces, right, environmental maybe forces, are going to act to change a population through changes in allele and genotypic frequencies. Okay, so that's where we're going. And we'll talk about, when we talk about population genetics, you're going to hear the term gene pool. And so what a gene pool is it's going to be a collection of all of the potential alleles for a particular gene that are present in a population. That's going to be the population's gene pool. And each organism will inherit 2 alleles out of that gene pool. So what we can do is we can quantify them. We can quantify allele frequencies, so we can put a number on them, a percentage on them, or a frequency, we can say, on them. And so you have a gene pool, which is all of the alleles in a population. And allele frequency is going to tell us what percentage or what frequency of the gene pool is a particular allele. Okay, it's the rate at which a specific allele appears in a given population. So here's an example before we get too deep into this. Let's say we're studying gene A. Okay, and so for gene A, there's 2 alleles in a population. There's the dominant allele, capital A, and there's the recessive allele, lowercase a. Now we have a population of 50 individuals. Okay, so if we do the math, if we've got 50 individuals and each person is diploid, then there's going to be a total of 100 alleles present in that population. Let's say that 75 of those 100 alleles are dominant, capital A alleles, and 25 of those alleles are the recessive form, lowercase a. If I asked you the frequency of the dominant allele, you would just take 75 divided by 100, and that would be 0.75, or 75% of the alleles are dominant. If I asked you for the frequency of the recessive allele, lowercase a, you would just take 25 divided by 100, and you would tell me that the frequency of the recessive allele is 0.25, or 25%. Okay, so we can put a number on it. We can say, okay, of this gene pool, this is how many of this allele, or this is how many of this allele that I have in the population. Okay, so before we get any further into that, I want to just remind you again that natural selection is going to select for those phenotypes that are going to make the organism better suited in that particular environment. So we call those adaptive phenotypes. So natural selection is going to select for individuals that have a more adaptive phenotype. And therefore, it's really selecting for adaptive genotypes, right? Because phenotype is coded for by the genotype. And so, when natural selection operates, when you have natural selection happening, you're going to end up with a change in allele frequencies in a population over time. Okay, and that's what population geneticists are studying. They're studying changes in allele frequencies in a population. If there are no allele frequency changes, then we would say that that gene is not evolving. It's not under natural selection. But if we do see changes, then there is evolution occurring, microevolution, right? Okay, that's what this next statement says. So if there is, or sorry, if a population is evolving, right, there's going to be some changes in allele frequencies for genes that are under selective pressure. So if natural selection is happening, we get changes in allele frequency. And that's going to be called, again, microevolution. It's not the evolution of a totally new species, but we're changing allele frequencies slowly over time. Okay, and that eventually could lead to macroevolution. Okay, so I've been saying all this time that natural selection is the force behind evolution. And it is, it's the most influential force, but there actually might be other mechanisms at play. So we're going to come to some of this stuff towards the end of the chapter, but I just want to let you know from now that sometimes evolution is really due to chance. So we're going to talk about something called genetic drift. In genetic drift, we're going to talk about two types, the bottleneck effect and the founder effect. In both of those, they're both forms of genetic drift. What we're going to have is we're going to have changes in allele frequencies. So we're going to have microevolution happening in a population. However, when we look at the gene where we have this occurring, natural selection's not at play. So when we get this microevolution occurring, there isn't any adaptive advantage over pre-existing frequencies. So again, it's not natural selection at play, it's actually just due to chance. So one type of genetic drift is called the founder effect. So let's look at this really quickly. So with the founder effect, you have a population of organisms, right? And a small subset of that population, of that parent population, leaves for whatever reason. It gets isolated from the original parent population. So now you have this small population that just kind of branched out. It just became isolated. And that isolated population is probably only going to have a subset, a fraction of the alleles that were present in the original population. And therefore, off the bat, there's going to be an allele frequency change in that population. Nothing happened. There wasn't any selective force. They just happened to become isolated, and now reproductively, they're isolated as well. Okay, so again, we're going to talk more about that towards the end of the chapter. Okay, so I have to introduce you to Hardy-Weinberg principle of equilibrium. And so Hardy and Weinberg, these were two scientists that in the early 20th century, they were studying genetics. They were looking at populations, and they founded this principle. What does it state? It states that a population's allele, and therefore genotype frequencies, they're inherently stable, which means that allele frequencies, phenotype frequencies, genotype frequencies, they should stay the same over time, unless there is some type of an evolutionary force acting on the population. Unless something happened that would drive changes in those allele frequencies. Okay? If there is nothing driving those changes, then theoretically, those allele, phenotype, and genotype frequencies should remain constant over time. They shouldn't change. And so this is a great mathematical model to show frequencies. So we've got some letters to talk about here. We're going to use P and Q. When you see P, P is going to represent the frequency of the dominant allele in a population, and Q will represent the frequency of the recessive allele in the population. Now we've got two formulas that we're going to have to learn, and both of these formulas are going to be true when we're studying genes in which there's two alleles that exist in a given population. Okay, so we're going to keep things pretty simple. The first formula that you should know is that P plus Q is going to equal 1. So what does that mean? That means the sum of the frequency of the dominant allele and the recessive allele will always equal 1. You can use this formula if you're trying to figure out the frequency of one allele if you know the frequency of the other. So for example, if you know the frequency of P is 0.5, then what you would do is 1 minus P, right? So 1 minus 0.5 equals 0.5, so Q would equal 0.5. Okay, so if you know the frequency of one allele, you can figure out the frequency of the other by using this formula. The second formula that you're going to want to become familiar with is right down here. It's P squared plus 2PQ plus Q squared equals 1. Now in this formula, P squared is going to represent the frequency of the homozygous dominant individuals in a population. Okay, so now we're really looking at individuals, right? So P squared are the homozygous dominant, right? The big A, big A. 2PQ, the second term there, is going to represent the frequency of the heterozygote individuals, right? So those individuals who are big A, little a, let's say. You're also going to hear us refer to them as carriers. So individuals that have both a dominant and recessive allele can also be known as carriers. They're carrying that hidden recessive allele. And the third term there, Q squared, that's going to represent the frequency of the homozygous recessive individuals. So all those that are, little a, little a. So both of those formulas are going to become important. So you want to make sure you know P plus Q equals 1. P is the frequency of the dominant allele. Q is the frequency of the recessive allele. P squared plus 2PQ plus Q squared equals 1. And make sure you know the frequency of the homozygous dominant is P squared. The frequency of the carriers or heterozygotes is 2PQ. And the frequency of the homozygous recessive individuals is Q squared. Okay, so this is really easy to use those two formulas if you know numbers of alleles that are present in a population, but usually you don't know that. Usually when you're given a genetics problem, you don't know the allele frequencies. And so all you're really seeing is phenotype. And so based on phenotype, that's how you have to figure out allele frequencies. So what do you do in that situation? So a lot of times a question will start off with, this many people in a population have the recessive phenotype. And therefore, what you're going to have to do is you have to know that when you're given that recessive phenotype, the frequency, right, the number of individuals, you can figure out frequency, is going to be Q squared. So the number of recessive individuals in a population is that term, Q squared, right? That's the frequency of homozygous recessive individuals. And so if you have that number, right, you have to take the square root of that in order to solve for Q. We're going to do some problems. Here's one on the next slide to demonstrate that. Okay, so we're going to apply Hardy-Weinberg principle. And let's say, for example, we're looking at rock pocket mice. And you saw a video on these. You watched it for the last chapter, so you know a little bit about them. So the particular gene that we're going to be studying is going to be fur color. So we've got these rock pocket mice, and they can either be tan or black. Now, tan fur color is the dominant phenotype, and black fur color is the recessive phenotype. We've got a population of mice. There's 586 of them. Okay, and 230 of them are black. And so based on what I just gave you, take a minute, pause the tape here, and I want you to try to calculate genotype, phenotype, and allele frequencies. So let me give you an idea of how to start, give you a hint here. So if 230 mice out of 586 are black, if you do that fraction, 230 divided by 586, that's your Q squared. So you should write that. Q squared equals 230 divided by 586. And that's how you should start. So why? So black we said is a recessive, right? And so the number here this is the number of homozygous recessive mice. Okay, total mice is 586. So again, if you do that division, that's going to give you Q squared. So start there. And again, pause this so you can do it. So after you finished it, let's check your work. And so again, at the very top here, what I was given, 230 black mice out of 586 total mice, that represents Q squared, the number of homozygous recessive divided by the total number of mice. And so if you do that math, you should have gotten 0.39. So Q squared equals 0.39. So what is that? Again, that's the frequency of the homozygous recessive mice, both the phenotypic and genotypic frequency. Okay, so what can I learn from here? I can figure out Q, right? So I take the square root of both sides. And so the square root of Q squared is Q. And the square root of 0.39, if you do the math, is 0.62. So I just solved for Q. So Q equals 0.62. What is that? That's the frequency of the recessive allele, the allele that codes for black in this case. So now if I know Q, I can figure out P. So now I'm going to use this formula. P plus Q equals 1. And so P is going to equal 1 minus Q, or 1 minus 0.62. That's going to give me 0.38. So now I know the frequency of my dominant allele, the allele that codes for tan. So what else can I figure out? I can figure out the frequency of homozygous dominant genotype, right? That's P squared. If I know P, if P is 0.38, if I square 0.38, I'm going to get 0.14. I can also figure out the frequency of the heterozygous genotype. And so that's just going to be 2 times P, which is 0.38, times Q, which is 0.62. That gives me the frequency of the heterozygous genotype. Okay, so I've got the frequency of both alleles. I've got the frequency of the recessive genotype and phenotype. And I've got the frequency of the dominant genotype and the heterozygous genotype. And the only thing left to calculate is the frequency of the dominant phenotype. So I've got to take both of these, my P squared, which was 0.14, and my 2PQ, which was 0.47, and that's going to give me 0.61. Okay? I could have also done 1 minus 0.39, again, the same thing. Either way is fine. So I just figured out genotype, phenotype, and allele frequencies based on that problem. Okay, and so what Hardy and Weinberg, what those equations really tell us, right, what their principle really tells us, is that if a population is at equilibrium, so if P and Q values stay the same over many generations, that's equilibrium. And therefore, the population, we would say, is non-evolving. So does that ever really happen? Yeah, there are some genetic loci that are not evolving, that we actually, if we look at them year after year, decade after decade, we find the same P and Q values, but that's not very common, okay? In the natural world, there's usually some kind of a selective pressure, right? There's usually, when we look at genetic loci or populations in general, we usually do have change over time, this microevolutionary change. So in the natural world, Hardy-Weinberg principle, this ideal situation that it puts forth, it really seldomly occurs. What's required in order to have Hardy-Weinberg equilibrium? Okay, so genetic equilibrium, it's only going to exist when all 5 of these conditions are met simultaneously. So you have to have all 5 of these happening. So the first is random mating. So mating within a population must be random in order to get Hardy-Weinberg equilibrium. Okay, there can be no selective pressure in mating. We're going to learn a little bit later in the chapter that that's, very rarely the case. There's usually mating is going to be non-random. Okay, secondly, there could be no genetic changes in the DNA, so no mutations, which again, that's a random event, so highly unlikely. So those are 2. The third is that the population has to remain large. There has to be a large sample size, a large population present. You can't have genetic drift. You can't have a bottleneck or the founder effect. Those kinds of things are going to go against Hardy-Weinberg equilibrium. We'll talk about those in more depth. The third is no migration. We're going to learn about gene flow in a couple of slides. You can't have individuals leave a population or come into, so immigrate or emigrate into a population. You can't have that in order to have Hardy-Weinberg equilibrium. And lastly, you can't have natural selection. So there could be no selective advantage for certain traits in a particular environment. You know, certain traits can't be adaptive. If you have that, then you're going to get evolution, microevolution. Okay, so before I go any further, I want you to pause it here. And I've got this slide of questions for you, goes along with your Are You With Me's. And then there are some genetics problems. And so I'm going to have you try those problems. And then I'm going to actually work through some of them as well. Okay, so pause this here. This slide you should be able to answer no problem. But the next couple of slides, I'm going to help you through them. Okay, so this genetics problem, we've got an eye disease is caused by a recessive allele. Okay, so that's the trait that we're looking at, is this eye disease, having it or not. And having the eye disease is recessive. Okay, so you've got one person in 100 that has the condition. So you've got, I'm going to try to write with this thing, 1 out of 100, one person out of 100 people. I'm going to do the typing thing next time. Equals Q squared. Okay, so if you do the math there, 1 divided by 100 is 0.01. And so then if you do So if you do 0.01, if you take the square root of that, oh, there's my daughter. So you're going to get Q. So Q is going to equal 0.1, right? So the square root of 0.01 is 0.1. So Q equals 0.1. Okay. Okay. So we know Q. And so now if you want to figure out P, P is going to equal 1 minus 0.1. So P equals 0.9. Right? Because P plus Q equals 1. Okay, so there's the first question. What are the values of P and Q in this population? The second question, what is the percentage of carriers in that population? So remember that carriers are going to be 2PQ. So 2PQ, there we go, I'm writing better now. So that's going to be 2 times 0.9 times 0.1. So let's do that. 2 times 0.9 times 0.1 equals 0.18. Now that's a frequency. To get a percentage, you got to multiply that by 100. So that's going to be 18%. Okay, so there's the first genetics problem solved for you. Okay, so let's go to the next problem. Okay, so we've got a rare and lethal disease that occurs when individuals are homozygous recessive for a particular gene. Okay, so homozygous recessive. Okay, so having this rare lethal disease is only going to be conferred by the homozygous recessive gene. The frequency of this condition occurs in 1 in a million individuals. Okay. So Q squared is going to equal 1 divided by 1 million. Okay, and there you're going to figure out Q. And we got a long number here. So Q is going to equal 0.000001. Oh, sorry, Q squared. Okay, and so now we got to take the square root of both sides. And so now we got to take the square root of both sides. And so now we got to take the square root of 0.000001. Okay, and so now we got to take the square root of both sides. And so now we got to take the square root of both sides. And so now we got to take the square root of both sides. And so the square root, which will give us q, I'm going to come up here, q equals 0.001. Okay, so we've got q from q squared. Okay, so the question is, how many individuals in a town of 1,400 can be expected to carry this allele? Carry. How many individuals? And we've got 1,400 total. So we're looking for the frequency of carriers. Okay, I know that because of that term carrier. And so once we have the frequency of the carriers, we can multiply that by the total population and get the number of carriers. So I have to figure out P, right? So P is going to equal 1 minus Q. So 1 minus 0.001 is equal to 0.999. And now we need to do 2PQ, right? It's my number of carriers. So 2 times 0.001 times 0.999 is equal to 0.001998. I can't fit this. Hold on, let me try to fit that a little bit better. Point 0.009 998. There. Now it fits in this in this green. Okay. So that's the frequency of carriers. All right. So now I have to take that number and I have to multiply it by the total population, which is 1,400. So times 0.00998. So it's about 3 people. So 2.79 people, which is about 3 individuals, are expected to carry this allele. Okay, so again, I started off, I was given 1 out of 1,000,000, and I know that that's the number of homozygous recessive, or the frequency of Q squared. Okay, so Q squared equals 1 divided by 1,000,000. And then if I take the square root of each side, I figured out that Q was 0.001. I can figure out P by doing 1 minus Q. So P equals 0.999. And then I had to figure out the frequency of carriers. So that's 2PQ. And once I got that number, I could take the frequency of carriers multiplied by the total number of people in that town, and I get the number of carriers, right? So about 3 people out of 1,400 will be heterozygous for that particular gene, and they would die, right? Because it's lethal. Okay. Next question. 4% of a population, here is my, okay, so 4% of a population suffers from a serious and debilitating recessive disease. Okay, so again, a recessive disease. And it's 4%. So what does that tell me? 4% is equal to 0.04. And that's going to be the frequency of my Q squared. Okay, so I know that I've got this recessive disease and that 4% of a population suffers from that. So that's Q squared. So right from that I can figure out Q, right? So if I take the square root of both sides, I figure out that Q is equal to 0.04 square root 0.2. And so then P That's an easy one, is equal to 0.8, right? Because P plus Q have to equal 1. The next says how many carriers are present in a population of 2,500. So I have to figure out carriers. So 2 times 0.8 times 0.2. Let's do that math. 2 times 0.8 times 0.2 equals 0.32. So that's the frequency of carriers. We want to know the number out of 2,500. So I have to do 2,500 times 0.32. That's going to give you the number of carriers. That's 800. So there's 800 carriers. So I just answered the first two questions. Let me erase some of this real quick. Okay, so we got 800 for this answer. Hold on, let's do this. So we don't forget, just in case you need that. And I'm going to erase. Ooh. Erase. Okay, I'm going to get my pen back. Okay, just like we're in class. Okay, so the next thing says, if two carriers had children, what are the probabilities of their having offspring with a serious condition? We're taking you back to chapter 12 here. So if we have two carriers, we can use any letter to represent this. I'm going to use the letter A. So that would be big A, little a. That's one carrier, mating with a second carrier, big A, little a. So what's the probability, what's the frequency or the percent of their having offspring with a serious condition? So if you did this, if you did the Punnett square, hopefully just by looking, you'll know that 1/4 or 25% of the offspring are going to be little a, homozygous recessive, and therefore suffer from this debilitating recessive disease. Okay, last part of the question, it says, after repeated generations of selection, would you expect Q to drop to 0? Why or why not? And so what you hopefully have seen is that non-adaptive alleles, right? And so obviously an allele that would code for a serious debilitating disease is a bad allele, one that's not adaptive. That's going to be selected against. However, that recessive allele is hidden in heterozygotes. And so it's only going to be selected against in the homozygous recessive form. And so that question of will Q ever drop to 0 is a confounding question. It's hard to answer. And the answer is probably not. And that is because it is hidden in heterozygotes. So it's not that it definitely won't get to 0, but because it's hidden, it's probably going to be maintained in the population. Okay. So I think, yeah, this is the last one. This one's tricky. Okay, so we have 2 men and 3 women, and they settle on an uninhabited tropical island. All 5 settlers had brown eyes. Okay, so you've got 5 people here. This is a great example of the founder effect. The allele frequencies that this population is going to have at this point is going to be very different from the original population because there's only a few of them that have left the main population. So you've got 2 men and 3 women. They're going to leave original parent population and they're going to settle on an uninhabited tropical island. So they're alone. And all 5 of them have brown eyes. Brown is dominant to blue, just like usual, right? One of the men carries a recessive allele for blue eyes, while the rest are homozygous brown. Oh, this is interesting. This actually tells us alleles. So we don't have to go to Q squared, right? There are none that are homozygous recessive, so we don't even know that. We actually know the alleles. So if we have 2 men and 3 women, we've got 5 people, and so we have 10 total alleles in that original population. I'm going to need another sheet of paper for this one. Okay, so we have 10 alleles. And if we take a look here, one man carries the recessive for blue eyes. Okay, so only one allele out of 10 that's going to be the blue allele. So this is going to be easy. So Q equals 1 divided by 10, or 0.1, and P is going to equal 9 divided by 10, or we could do 1 minus Q either way. So that's 0.9. Okay, so there's the first one. 0.9 is P, and Q is 0.1. Okay. Okay, second question says, the population grows to 1,500 individuals and is in Hardy-Weinberg equilibrium. So what does that mean? That means P and Q values have not changed. How many blue-eyed individuals would there be in this expanded population? So to be blue-eyed, you have to be Q squared. So Q squared equals 0.1, right, squared. And so Q is going to equal 0.0. Oh, what am I doing? That's not what I meant. Q squared equals 0.01. Okay, so that's the frequency of blue-eyed individuals. So there's 500 people. So how many of them are blue-eyed? Sorry, 1,500 people. 1,500 times Q squared. That's going to be 15. So your answer to this one, how many blue-eyed individuals, is 15. Okay. All right, let me erase this so I have room. The next question says, how many brown-eyed individuals? So I got to get P squared. So P is 0.9. So if I square it, that's going to give me 0.81 times 1,500. Oh no, excuse me. I'm forgetting. Brown-eyed individuals can be both P squared and 2PQ. So hold on a minute. Almost screwed that up. So 0.18, but we also have 2PQ. Okay, so 2 times 0.9 times 0.1, 2 times 0.9 times 0.1 equals 0.18. So we have to add these. So 0.18 plus 0.81 equals 0.99. Okay. So 0.99 times 1,500 equals 1,485 people have brown eyes. Now you could have taken the easy way out and you could have said, well, if 15 have blue, the rest have brown, so 1,500 minus 15, and you'd get the same answer, right? So either way is fine. I just wanted to practice using those terms. So we got 15 blue-eyed people and 1,485 brown-eyed people, both the homozygous dominant as well as the heterozygotes. Okay, let me erase this. Okay, next question says, this gets a little tricky. Okay, so it says, if all of the blue-eyed individuals died at the same time, so all 15 of them died, what would be the new P and Q values of the following generation? Okay, so first off, this is what you would be left with. Okay, and 1485 times 2. So that means you've got 1,485 people. If you multiply that by 2, in that population you have 2,970 alleles. Okay, alleles. let's go back to the original population of 1500. So in that population, you had 1,500 individuals. So originally you had 3,000 alleles. Okay? And so let's think about this. If 15 blue-eyed people die, you've lost blue alleles, but you haven't lost any brown alleles, right? And so if you can figure out how many alleles in that original population of 3,000 alleles were brown, dominant alleles, then you can figure out the frequency now in this population, because the frequency's changed, but the actual number of dominant alleles has not. So if I go back to my original population, 1,500, that's 3,000 alleles. And I multiply that by the frequency of the dominant allele, 0.9, right? That was the original p-value. So let's do that. Ooh, hold on, I did something wrong. 3,000 times 0.9, 2,700. Okay, so there were 2,700 dominant alleles. And we know that there's still 2,700 dominant alleles because nobody died that had that allele. So if I divide the number of dominant alleles by the total number of alleles that are now present, right, that's what's left over here. We lost 15 people. So let me do that math. 2,700 divided by 2,970 equals, that's my new p, 0.9, well, let's round up, 1. That's p. And so q would be 1 minus 0.91, 0.09. So there was a slight shift. Okay? So yeah, you got to go to alleles, and you have to remember that if you lose all of the homozygous recessive individuals, then you've only lost recessive alleles. Let's think about the opposite of that. What if I said to you, if all of the brown-eyed individuals died at the same time, what would be the new P and Q values of the following generation? And so there you'd say, well, I lost everybody who was brown. I have no dominant alleles left. All I have left are recessive alleles. And so P would become 0 and Q must be 1. Okay, so moving on from population genetics problems, we are going to spend the next part of this chapter talking about what happens if we don't follow Hardy-Weinberg equilibrium. We know that microevolution happens, so we're going to talk about some of those situations that would lead to that. Okay, so we know that most often, individuals in a population are going to display different phenotypes, right? Unless there's only one allele for a particular gene. However, usually there's multiple alleles, sometimes 2 or more. We call these differences in alleles, we call them polymorphisms. Okay, so when there's different alleles or subsets of alleles that are expressed for a particular gene, those are called polymorphisms. And so a gene that has multiple alleles would be a polymorphic gene. There's more than one phenotype that can be produced. When you have a polymorphic population, again, you're going to have 2 or more variations or alleles for a given gene. And therefore, again, you're going to see a polymorphic genetic profile for those individuals. Another term that hopefully you identified was population variation. This is used to describe the distribution of different phenotypes among individuals in a population. So there's variation, right? Remember, variation is going to be the thing that natural selection is going to act upon. There has to be variation in a population in order to get evolution. This variation is, of course, influenced by genetics, but the environment does also play a role, so we have to keep that in mind. And so we can actually calculate how much of that variation is due to genetics and how much is due to the environment. And that's going to be key to predicting future evolution for that particular gene, that particular population. Okay, so again, that was population variation. That's the distribution of phenotypes among individuals in a population. And genetic variance, that's the genetic differences in a population. So again, that can be measured. And those genetic differences will be either selected for or against, depending if they're adaptive or not, when natural selection acts upon phenotype. So even though we always say that natural selection will select for a particular phenotype, it's also selecting for a genotype. Okay? Remember that evolutionary forces that play in natural selection, they're only going to act on heritable traits. So only those that are genetic and only those that are passed from parent to offspring. Any traits that are due to the environment, how the environment is playing a role there, or any traits that are acquired during an individual's lifetime, those don't work here, right? So those would not be genetic, and therefore they don't get passed from parent to offspring. Remember the Lamarck's hypothesis of the giraffe necks disuse hypothesis? That doesn't come into play. That natural selection is not going to act upon those types of things, right? So heritability, that's going to be the fraction of a phenotypic or a phenotype variation that is due to genetic differences. So when we talk about heritability, we're talking about just those traits that make up phenotype that can be passed from parent to offspring. And lastly here, just want to make sure that you guys are aware, I think that we've already said this, we talked about it even last semester, General Biology 1, the greater the genetic variation, right? So the greater the phenotypic diversity within a population, the better chances there are for survival of that particular species. Your textbook talks about breeding programs at zoos, and this is really important. Zoos are able to mate in captivity organisms that are either endangered or they have the possibility of becoming endangered and extinct. And these breeding programs at zoos are able to really selectively breed, so it's really artificial selection, and they can select for phenotypic variation that's really favorable for the species. And then there are some breeding programs that will put these organisms back out into the wild. But when we lose a species because of a lack of genetic diversity, again, sometimes those programs can actually bring back a species. There's been some examples of that, so it's pretty cool. I want to note here, what can lead to a decrease in genetic variation or a decrease in genetic variation. And one of the things that can lead to this is inbreeding. Inbreeding, for the most part, is not a favorable thing, right? But we do see this in some species. And sometimes when inbreeding occurs, again, you're going to get a decrease in genetic variation. And we get what's called an inbreeding depression. This is a phenomenon that's observed, again, when inbreeding is taking place in a population. And what happens is you end up with a lower fitness. So there's these bad, deleterious outcomes, this lower fitness. It's going to increase the likelihood of those relatively rare recessive diseases. They're going to pop up more often than not. If there wasn't inbreeding, then those rare recessive diseases would be selected against or lost. But when you have inbreeding, there's a higher likelihood to get those rare diseases that pop up. And again, you get a decrease in genetic variation. So inbreeding is not considered to be a good thing. Okay. So again, we mentioned this earlier in the lecture. We know that natural selection is the main evolutionary force, right? That's acting here, that's acting to drive evolution. However, there are other forces at play. And again, we alluded to some of these, but we're just going to spend a little bit of time on each of these. So genetic drift, gene flow, mutation, and non-random mating. All of these things go against Hardy-Weinberg principle, And therefore, if these things are at play, then we will get microevolution. Okay, so let's talk about genetic drift first. So genetic drift is evolution by chance. Okay, so natural selection really isn't playing a role here. So for whatever reason, and we'll talk about two different examples, it's by chance some individuals in a given population are going to have higher reproductive success than others. There's no selective pressure and there's no reason behind this. Instead, again, it's due to chance. So when you think of genetic drift, I want you to think chance. Again, it's still going to result in evolution, changes in allele frequencies, because you have differences in reproductive success. There's just not a driving force or a reason behind that. There's no adaptive trait that's being selected for in this case. Genetic drift is much more common in smaller populations. So remember, that goes against Hardy-Weinberg's principle. You have to always have a large population. Well, if you have a small population, you are going to get changes. And the reason why we think this is because the gene pool is smaller in a smaller population. Okay, genetic drift can be magnified by natural events like natural disasters that take a large population of individuals and the natural disaster hits and a large portion of that population dies. Okay, so what are you left with? A smaller population. If you take a look at allele frequencies of the original population and compare it to the population that remains, there's definitely going to be changes in allele frequencies, right? There was no selective pressure here. There was a natural event that happened due to chance and therefore changed allele frequencies of the population, right? So we have two examples. So when we have a natural disaster, we're left with something, my pointer's not working. We're left with something called, oh, there it is, the bottleneck effect. Okay, so if you look at the bottle over here, right, you see all these beads, all these colored beads, that would represent people in the original population, and the different colors represent variation in whatever gene we're looking at. And so some natural disaster happens, maybe it's a hurricane or coronavirus, something. And we get a bottleneck. And so what does that mean? Means only some of the individuals, right, the individuals that are able to escape out of the top of this bottle end up in this glass, right? And those are the only people that survive. And so we take a population that had a lot of variation in it, and we end up with a population that has less variation in it. So again, that's called the bottleneck effect. So again, you have a large population and only some of them survive. When you look at the surviving population, again, allele frequencies, P and Q values, are going to be different than the original population. Okay, why? Because we decreased in size. We've got less alleles present in the gene pool. The second type of genetic drift that I just want to briefly talk about is the founder effect. And again, we did a genetics problem that actually displayed this. So the founder effect is when you have a large population again, and a small number of those people, or animals or seeds, whatever type of organism we're talking about, leaves the original parent population and settles in a new area, a new location, right? Oftentimes, separated by some type of a physical barrier. That's going to lead to changes in allele frequencies. So if you look at the founder population, the new population, the genetic structure, the allele frequencies, the gene pool is going to look different than the original parent population. And so yes, you're going to have evolution, microevolution, and yes, you're going to have changes in allele frequency. So genetic drift goes against Hardy-Weinberg equilibrium, and it's definitely going to lead to changes in allele frequencies. So you've got genetic drift, including the bottleneck and the founder effect. Okay. The second here that's going to go against Hardy-Weinberg equilibrium is gene flow. Okay, so remember, Hardy-Weinberg equilibrium said no one can come in or leave a population in order for that population to remain at stasis, right? Stable, not evolving. Well, with gene flow, you are going to have immigration or emigration. So right here in this beetle population, we've got this subset of beetles here that's green, and we've got a separate population, I don't know, maybe down the road or lower on a tree, that is going to be phenotypically different, right? And so this is a different, maybe a different species. Or maybe it's the same species, just two different populations of that species, just two different colors. So if this brown beetle, decides, hey, I'm going to go visit these guys, and then ends up reproducing with them, well, now you've got the flow of not just that organism, but their genes. Okay, so gene flow. That's going to be the flow of alleles into and out of a population due to migration of individuals or migration of gametes. That's going to lead to evolution. Okay, mutations, we've already talked about this. Mutations are going to be any changes to an organism's DNA. Mutations increase genetic diversity. Remember, that's the ultimate source of all diversity. Evolution of a new species is actually going to result from the accumulation of mutations over time. Harmful mutations will be selected against, but beneficial mutations will be selected for. Okay, and so again, if we think back to Hardy-Weinberg principle, they told us that mutation cannot happen in order to stay at evolutionary stasis. And so if it does, we're going to get evolution. We're going to get microevolution. Okay, so next, non-random mating. So remember that Hardy and Weinberg told us that mating would have to be random in order to stay at evolutionary stasis. And so if mating is not random, then we're going to get evolution. This is a typo. So positive assortative mating, that should say positive before that. That's the selection of mates with similar phenotypes. And there's actually the opposite of that called negative assortative mating. That's selection of mates with dissimilar phenotypes. So there can actually be both of those. So again, that's non-random mating, right? It's selecting mates that look like you or selecting mates that don't look like you. In both cases, it's not random mating. And second here, sexual selection. This is the selection of mates based on behavior or phenotype. Okay, so assortative, positive and negative assortative are forms of sexual selection. Oftentimes, when we're talking about a species or populations of species, non-random mating is going to occur purely because of physical location. How close two organisms live to each other, how much time they spend near each other. Right? So if a population is large and individuals are kind of spread out in an area, organisms that spend more time near each other are more likely to mate than organisms that spend less time near each other. One negative of non-random mating is inbreeding. Again, organisms that live close to each other have a tendency, maybe not humans, but other organisms, to breed with each other even if they are closely related. And so that can be bad. That can lead to that inbreeding depression, the expression or the inheritance of bad deleterious traits. Okay, so take a break here and answer these questions. When you're finished, you can go on. Okay, so I have a couple of questions for you before you move on. You'll notice that I didn't put questions after the first Are You With Me because I thought that we had enough genetics problems there. So here's some questions for our second Are You With Me? Okay, so the first one, population genetics is the study of how selective forces change the allele frequency of a population over time? Or is it the study of the genetic basis of population-wide traits, the study of whether traits have a genetic basis, or the study of the degree of inbreeding in a population? So which one of those is the definition for population genetics? Okay, hopefully you chose the first answer here. How selective forces like natural selection or genetic drift or the founder effect, the bottleneck effect, any of those, how those forces change or alter allele frequencies in a population over time. That's population genetics. Second question, which of the following populations is not in Hardy-Weinberg equilibrium? So this takes us back to that first section. So hopefully you understand Hardy-Weinberg equilibrium describes a population that's in stasis, so not evolving. So which one of these describes a population not in Hardy-Weinberg equilibrium? A population with 12 homozygous recessive individuals, 8 homozygous dominant individuals, and 4 heterozygous individuals. would that be a population that's evolving and not in Hardy-Weinberg equilibrium? Blue, a population in which the allele frequencies do not change over time. Does that violate Hardy-Weinberg equilibrium? Or the formula, p squared plus 2pq plus q squared equals 1, does that represent a population not in Hardy-Weinberg equilibrium? Or yellow, a population undergoing natural selection? So again, which one of those describes a population that is not in Hardy-Weinberg equilibrium? Hopefully you chose yellow, a population undergoing natural selection. Okay, so remember those characteristics which are going to tell us if a population is evolving or undergoing allele frequency changes, so not in Hardy-Weinberg equilibrium. One of those is natural selection. Okay. Next question. One of the original Amish colonies rose from a ship of colonists that came from Europe. The ship's captain, who had polydactyly is extra digits. It could be extra fingers or extra toes. Polydactyly is a rare, dominant trait. The ship's captain, who's one of the original colonists, had polydactyly. Today, we see a much higher frequency of polydactyly in the Amish population when we compare it to the general population of the United States. Okay, so polydactyly is more common in the Amish population. This is an example of, is it an example of natural selection, the bottleneck effect, founder effect, or inbreeding depression? So think about that for a minute. You've got an original colonist, or original group of colonists that came over from Europe and set up a colony in the United States where you have a higher frequency of this rare, dominant genetic condition. Hopefully you chose founder effect. Okay, so we have a small population who leaves the parent population and colonizes a new land. They're going to take with them only a fraction of the alleles. Okay, and in this case, the allele that codes for having extra digits. So again, that's founder effect. Oh. Sorry about that. Okay, so next one. When male lions reach sexual maturity, they leave their group in search of a new pride. This can alter the allele frequencies of the population through which of the following mechanisms? Would we call this natural selection, the bottleneck effect, the founder effect, or gene flow? So you have an individual in a population who's now going to leave and take their alleles with them and go to a new population of that species. And who knows, they might come back, they might not come back. So what do we call that? We call that gene flow. Okay, when you have alleles flowing between two or more populations. Okay, so that's gene flow. Next question, yep, this is the last one. Which of the following evolutionary forces can introduce new genetic variation into a population? So new genetic variation. So the first one is natural selection and genetic drift, mutation and gene flow, natural selection and non-random mating, or yellow, mutation and genetic drift. Think about that for a minute. So which of the following can introduce new genetic variation into a population? So the best answer here would be in blue. So mutation, right, that's the source of all new genetic variation. And then gene flow. If you have an individual of a population leaving one population and going to another, they're going to possibly introduce new alleles, new genetic variation into that population which they are going to be entering. Okay, so go on and look at the next part, and then I think we just have one section left. And then after that, I'll have a couple more questions for you. Okay, so we're at the tail end of the chapter here. A couple of other things to talk about. Adaptive evolution, right? So natural selection will select individuals that have those traits that are most adaptive in that particular environment. So those phenotypes will be selected for, and those organisms will then probably reproduce, right? They're the organisms that are going to make it to reproductive age. So when we talk about fitness, that's what we're talking about. We're talking about those individuals that are most adaptive to that particular environment. And when we look at allele frequencies, so population geneticists, when they look at the actual allele frequencies, they're going to reflect that selection process. So population geneticists, they will look at forces driving those microevolutionary changes, and they should reflect each other. Now, selection, natural selection, can be stabilizing, directional, diversifying, or frequency dependent. So I'm going to spend the last part of this chapter going over those different types of selection. Okay, let me move this over so you can see that. Okay, got it. Okay, so the first one here, stabilizing selection. This is natural selection that's going to favor the average phenotype, and it will select against extreme variations of that phenotype. Let's go here to the middle. Students tend to be most intrigued by human things, right? And so I'm just going to get my laser pointer. And so in this figure right here, which is out of a past textbook that I used to use, this looks at human body weight, human newborn body weight. And on the x-axis here, we've got body weight in pounds. And then we've got actually two different y-axes. So this side here is going to be the percentage of the infant population in blue that survives. And then on this side, we've got the percentage of infant death. And that's going to be represented by the red line. So let's look at the blue line first. So if we look at infant weight, there's going to be a high mortality, which we can look at red too, right? So low blue, high red. If you have a baby that's born, less than a pound to 2 to 3 pounds, and as we increase in body weight, the chances of survival and the percentage of infants in the population that live, it's going to spike around 7 pounds, right? So the highest likelihood for survival, less likelihood of mortality is a 7-pound baby. If you go higher than that, that's going to also increase the risk of death. Okay. So again, this is stabilizing selection. So if we look, the average phenotype, around 7 pounds, is going to be selected for, right? These individuals will live. And if we get to the extreme down here, low birth weight, or up here, very high birth weight, those individuals will be selected against. Okay, so average birth weight in humans is around 7 pounds. Let's look at some other examples. Here's one out of your textbook. Okay, so this is dealing with robins. And so what you have here is a nest with these nice blue robin eggs. So robins typically lay a clutch, that's a collection of eggs, of about 4 eggs. Okay, anything higher than that, so more eggs, would result in malnourished chicks, and so they would not make it. And smaller clutches, you could be left with no offspring. So anything less than 4 is not good. So the average, what's been selected for by natural selection, is an average clutch size of about 4 eggs. Okay. Over here, we've got just kind of a make-believe beetle population, and what we're looking at is shell color. Okay, and it seems in this picture that beetles that have kind of a medium shell color have the highest likelihood of survival, where if you're a very light colored beetle, you have a low chance of survival, and a very dark beetle, again, same thing, low chance of survival. And so in this particular environment, whatever the environment is, being of medium light shell color is going to be selected for. So again, the average gets selected for and the extremes are selected against. Okay, so this is called directional selection. So different from stabilizing selection. In directional selection, we're going to select phenotypes at one end of the variation spectrum. Okay, let me take you to the beetles first, kind of like the first, just like the first slide. So the same thing, we have different colored beetles. We've got light, medium, and then we get darker as we go along. And so in directional selection, there will be some selective pressure. In this case, that's going to be selecting for darker colored beetles, so we get a shift in that direction, where we get an increase now in darker colored beetles while getting a decrease in lighter colored beetles. That's directional. One end of the spectrum, darker colored beetles, is going to be selected for over the other end of the spectrum, light. Here's another example. Again, this is from your textbook. This is a pretty famous example. You may have heard of it. This is the peppered moth. So if we look here, this is what the peppered moth used to look like. Okay, so we're looking at color. This light color. These peppered moths were around before the Industrial Revolution. And if we take a look at them, again, they're light in color, and they lived in a clean environment, right? Not a lot of, greenhouse gases or pollution was being produced at that time, and so the environment was clean. And so being a peppered moth, lighter in color, helped to camouflage them. Darker peppered moths, in this case, are what resulted after the Industrial Revolution. After the Industrial Revolution, we were left with an environment that was a little bit dirty. And so being a darker moth in a less pristine, less clean environment, kind of a sooty environment, was going to be selected for. It was favored because they were better to blend in with the environment that they lived in. Okay, so that's a great example. It's oftentimes used of directional selection. So this is where the population was to begin with. The environment changed after the Industrial Revolution. It became kind of sooty and polluted, and therefore we had this shift in our curve over here. Being a darker colored moth was more adaptive, more advantageous. So again, that's directional selection. The next one is called diversifying selection. This is when two or more phenotypes, usually towards the extremes of our bell, are going to be selected for by natural selection simultaneously. And the phenotypes towards the middle are going to be selected against. Okay, so let's go back to our beetles since we're familiar with those. Again, we have different shades of beetle color: light, medium, darker, darkest. And so something happens to the environment, and being a middle of the road colored beetle is not so great anymore. These are selected against. And so what is selected for is being light or being very dark. So that's diversifying. Okay, the two ends of the spectrum are selected for simultaneously. Here's another example from your textbook. This is a hypothetical rabbit population. We've got gray rabbits, we've got white rabbits, and we've got Himalayan rabbits. These are white and gray. And it turns out that in the original population, white rabbits were favored, but something happened in the environment, or these guys actually changed environment, I think. Yep, they ended up in rocky environments. And so being a white rabbit, they stuck out like sore thumbs, and they were definitely preyed upon a little bit easier. And so these guys decreased in frequency, and there was an increase in the gray and Himalayan rabbits. Okay, so the two ends of the spectrum were selected for. Okay, this last one is called frequency-dependent selection. And this can actually be positive or negative selection. So negative selection, this is the selection of phenotypes that are rarer or less common or present at a lower frequency. And positive selection selects for phenotypes that are more common or at a higher frequency. So in this particular example down here, this is from a past textbook that I used to use. This is an example of negative selection. And so what are we looking at here? So scientists sampled the numbers of cichlids. So these are cichlids right here, this one here, and this one here. And if we look at their mouths, These cichlids come in two different forms. They are of the same species. They can either have a right pointed mouth, and if they have a right pointed mouth, they're going to attack their prey on its left side, or their mouths can be left pointed. If they have a left pointed mouth, then they're going to attack their prey on the right side. And so what scientists did is over about a 10 or 12 year period here, from about 1982 to about 1990, a little bit farther than that, they went out and they looked at the frequency of cichlids with left pointing mouths. And it started off where the frequency of these left pointed mouthed cichlids was a little bit above 0.5. They come back a few years later and they find that it dropped. So they thought, ooh, those must be being selected against. And then all of a sudden it came back up. And then a couple years later it went back down. And it followed the same trend pattern. And so it's negative selection. So what happens when the frequency of the left pointing cichlids, left mouth pointed cichlids, increased, what happened is the prey, the fish, they learned that they were being attacked on the right hand side. And so they were able to learn to try to flee these cichlids. And so the frequency of these guys went down. They didn't have stuff to eat. And so the frequency of these guys now goes up until the fish learns that, oh, I'm being attacked on my left side. So they're more aware. And so the frequency of these guys goes down and this one goes back up. Okay, so the rarer phenotype gets selected for in this case. Okay, so here is our last Are You With Me? So you're going to talk about adaptive selection and then go over those 4 modes of natural selection that we just did. Just a couple more questions for you before we end this presentation. So there's our first one. So here we go. Finch beak size ranges from small to large. In the Grant's research on finches from last chapter, remember the video that we watched, we saw that following drought, finches with larger beaks were better equipped to crack open large seeds, and the population trended or shifted towards individuals with larger, deeper beaks. Remember, before that, there was kind of an even distribution with finches with medium-sized beaks probably being the most common. So then there was a shift towards finches with larger, deeper beaks. They were selected for by the environment. This is an example of, is this sexual selection, frequency-dependent selection, diversifying selection, or directional selection? And so hopefully you answered in yellow, directional selection. It was definitely a shift in one direction, which is directional. Okay, last one. In most plant species, medium height is selected for. Plants that grow too tall expend extra energy to do so and can get damaged by winds. Meanwhile, plants that are too short may not get the sunlight they need to grow. Okay, so plants that are of medium height are going to be, they're going to have an evolutionary advantage. So this is an example of, is this stabilizing selection, frequency-dependent selection, diversifying selection, or directional selection? So hopefully you answered stabilizing selection, right? So the phenotype in the middle is selected for. Okay, so that's the last question here. So you want to make sure, again, that you've got your vocab and your chapter outline. Those should have already been done before you did this lecture. And now your in-class assessment, your Are You With Me's, should also be finished. And you should upload those right to Blackboard. And then you are going to make sure you study, and then you're going to go ahead and you're going to take the quiz. Oh, before you take the quiz, you're going to also make sure that you do those population genetics problems. Those are also going to be due. And doing those before you take the quiz will help you in studying as well. Okay.